Today, I want to talk to you about the future of science at USANA. But I would like to begin with the past, specifically my past. As many of you know, I was born and grew up on the plains of North Dakota, a wide open land of farming and livestock. In my early years, I was surrounded not by a city or even a subdivision of residential housing, but the small town of Napoleon, only a few hundred people. In time, I became very aware of the presence of the fundamentals of life. For example, too many pesticides and herbicides in the air, water, and soil being taken up by plants and into the bodies of animals and people. I could not help but feel that things were no longer what they should be when I lost both of my parents while I was still young. My father to heart disease and my mother to cancer. They should have lived longer lives, but fate took them too early. This loss was a major reason for my decision to devote my life to research in medicine. I became aware that too many people were dying too young or were disabled too soon in life. I committed myself to doing everything I could to help prevent illness and disease what had so damaged my family. I chose to begin my medical career with microbiology. The greatest killers in the first half of the 20th century were diseases caused by infectious agents, bacteria, viruses, other microbes. It was, as they say, where the action was. The epidemic of Spanish flu that swept the globe in the second decade of the 20th century is probably the best example of how destructive infectious diseases can be. This pandemic infected 500 million people around the world, including remote Pacific islands and the Arctic. It killed 50 to 100 million of them, 3 to 5% of the world's population, making it one of the deadliest natural disasters in history. Fortunately, for later generations, following decades, science made tremendous strides when it came to understanding, preventing, and protecting against infectious disease. As a young man, I wanted to make the kind of contribution to the welfare of the human race which scientists like Louis Pasteur, Jonas Salk, or Linus Pauling did. That's why I worked for degrees in immunology and microbiology, and then founded a company to develop cutting-edge laboratory assays for infectious diseases. However, as the years passed, it became apparent that there was a major shift in the causes of disease and death. As the infectious diseases were being brought under control through improved diagnostic techniques, advances in sanitation, and the discovery of penicillin and other antibiotics, the incidence of another category of illness, the chronic degenerative diseases, steadily rose. The infectious diseases, in most cases, are what we call acute diseases, in which a diagnosis is made quickly based on one or two dominant symptoms. Then a drug or a procedure is prescribed to avert an immediate threat, such as serious infection or traumatic event that is beyond the patient's control. The mode of action is still essential for effective emergency and hospital care. In contrast, the chronic degenerative diseases, especially the metabolic diseases, are caused by complex, multiple conditions affecting multiple organs and systems in the body, leading to cellular degeneration. By definition, they are persistent. Treating symptoms may provide temporary relief, but the underlying causes are allowed to fester and perhaps worsen. Sadly, there is no question that the large majority of chronic disease is avoidable through nothing more than lifestyle modifications. Just common sense practices, such as following a healthy diet, getting regular exercise along with fresh air and water. In many cases, that is all it takes, especially if habits of healthy living are formulated at a young age. But now, early in the new century, chronic degenerative disease is at epidemic proportions. One of the most important reasons why the incidence of degenerative disease has grown so rapidly is that our current medical system is still using the same treatment methods developed in the first half of the 20th century. In many medical schools, students are still being taught to focus on the acute care model, despite chronic disease today being responsible for 78% of healthcare expenses and 70 to 90% of all mortality. To be successful in reducing the amount of suffering in the world due to disease, we need to abandon the model of acute healthcare that is accepted by medicine today. We instead need to replace it with a new approach to chronic degenerative disease, one that focuses on environmental and lifestyle factors, not acute infection and trauma. We know that heart disease, stroke, cancer, diabetes, and arthritis are not caused by any single agent, but by a combination of factors in our sedentary lifestyle, bad health habits, and exposure to an environment of toxic substances. science is now uncovering knowledge and developing new tools that are our best hope for dealing with the stories of planet's delicate disease. While certain aspects of this major shift in thinking first began to appear a couple decades ago, for example, the type of whole-body medicine practice at USANA Medical Institute, I think that we can say that the development of this new approach to healthcare was accelerated by the decision in the year 2001 to sequence the entire human genome, the Human Genome Project. The main goals of the project were to provide a complete and accurate sequence of the 3 billion base pairs that make up the human genome and to identify all the human genes. The primary purpose was to gain understanding of our genetic makeup so new medical treatments could be developed. Well, there were some surprises. It's known that the human body uses approximately 100,000 different proteins. So it was assumed there would be a similar number of protein-coding genes, one for each protein. But it turned out that we have only about 24,000 genes that code for proteins. Therefore, our one gene, one protein hypothesis was incorrect. Our genetic network is much more sophisticated and flexible than we expected. Second, there turned out to be much more duplication of some genes than expected, especially in sequences that initially didn't appear to produce proteins for cellular function. We are now rapidly learning how to make sense of this new knowledge. Our genome, the total of all the genes that make us human, are not a static library or a fixed blueprint. Genes can respond differently when they receive different messages from the environment. Many gene sequences that at first appeared not to do much at all are seen to play important roles in gene regulation and expression, turning other genes or groups of genes on or off. When the structure of DNA was first discovered, we considered it